Good morning, students. In this part, we will talk about the female reproductive part of the flower, and we will also understand the structure of Megaspore, that is ovule. Gynoecium represent the female reproductive part of the flower. Gynoecium may consist of a single pistil or may have more than one pistil. If there is single pistil called monocarpellary. If more than one pistil, called multicarpellary. For this, two terms are applied. One is syncarpus, other word is apocarpus. Syn means if they are fused together. Syn means fuse. Apo means free. If they are free, then it is called apocarpus. Let us talk about the pistil. Pistil consists of stigma, style, and ovary. This is the stigma. This is the part. In this part, the pollen grains gets attached after pollination. This one is the style. Style is the cylindrical structure through which pollen tube moves downward towards the ovary. It may be hollow or solid. This one is ovary, which is placed in thalamus, attached with thalamus. Ovary contains ovule, or inside the ovule, embryo sac or female gametophyte present. So, let us talk about the detailed structure of ovule. So, we are drawing a structure of ovule which is more common in angiospermic plant. This is our integument. This is funicle, this is ovary wall, this is the placenta. This is funicle. At the junction of funicle and ovule body is the hilum. This is outer integument, inner integument. This is bitegmic. Two integument present. This integument later on converted into the seed coat. In this central portion, the mass of cells, this mass of cells, this is called nucleus. This is called nucleus. Remember, all the structure is sporophyte, because meiosis has not taken place here. After the meiosis, the gametophytic generation will obtain. This is the micropyle. This is the chalaza pole. Micropylar pole. The ovule is attached to the placenta through funicle. Hilum is the point of attachment through which body of the ovule attached with funicle. Now we will try to understand the process by which the female gametophyte is formed. This nuclear cells, which is towards the micropylar end, selected for the meiosis. Remember, before the meiosis, all the parts, whether the nuclear cell, integument, hilum, funicle, placenta, all are diploid and all are in sporophytic phase. After the meiosis, the plant enters into the gametophytic phase. So, this cell selected for the meiosis. Now, we start calling this cell, this is diploid, we call starting this cell Megaspore mother cell. The Megaspore mother cell undergo meiosis. When meiosis first takes place, it forms two cells. After the meiosis second, that is division 2, 4 cells form. All are haploid. Out of these 4 cells, 3 degenerate. 3 degenerate. Out of 4 megaspores, 3 degenerate and 1 survive. This surviving megaspore, we are drawing here. This surviving megaspore forms embryo sac, will form the embryo sac after 3 mitotic division. Remember, first karyokinesis will take place, later on cytokinesis occurs. First mitotic division, that is division 3, this is the division 1, this is division 2, this is the division 3, this is mitotic. Karyokinesis, 2 nuclei will be formed. Now, again mitotic division, that is karyokinesis, division 4, 4 nuclei form. Now again 5th division, that is 3rd mitotic division. These two are the meiosis first and meiosis second. This is the first mitotic, second mitotic and 3rd mitotic, karyokinesis only. 8 nuclei forms. This is the 8 nucleated embryo sac. Out of this 8 nuclei, 3 migrate to one pole. Remember, this is the chalazal end or this is the micropylar end. and 3 migrate to this end, that is macropolar end, and 2 remaining is the polar nuclei in the center. This one is the 3 nuclei to one pole, these 3 nuclei in this pole, that is macropolar end, this is the chelazal end, and 2 nuclei in the center. Now cytokinesis takes place. Now cytokinesis takes place. 6 of the 8 nuclei surrounded by the cell wall, that is cytokinesis takes place in the around the 6 nuclei. 6 nuclei, here cell wall formation takes place, this cell wall formation 3. Out of 8, 6 nuclei surrounded by the cell wall. These 3 cells, the remaining 2 nuclei in the center is called polar nuclei. These 3 cells are called antipodal cell. And these 2 called synergids. One is the egg cell. This is a complete embryo sac. Now count the nuclei. 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei. And count the cell. Count the cell. 1, 2, 3, 4, 5, 6. One is the central cell. 7 cell. So this embryo sac is 8 nucleate and 7 cell. This is the characteristic of angiospermic embryo sac. This embryo sac is more common. This is called polygonal type. This is called polygonal type of embryo sac, or this is monosporic. This is monosporic because single megaspores forms this embryo sac. That is why it is monosporic. So this is the megagametogenesis. This is the megagametogenesis. That is, megagametogenesis is formation of development of female gametophyte. Development of this female gametophyte is called megagametogenesis.